Good afternoon, everybody, and hopefully you can see my title slide now. So what I want to talk to you this afternoon about is using computers and AI to design future medicines, and we've titled that Designer Drugs. So, who am I? My name's Stephen Doughty. I'm the head of pharmacy. You'll see some words there, Pennaeth Rhaglen Fferythiaeth. That's because we are in Wales, in North Wales, here at Bangor University. Hopefully, if you can see me, you can see a picture behind me of the wonderful North Wales scenery. And if you haven't visited North Wales, you really should come and see our university and see the wonderful setting that we're in. But that's the only bit of Welsh that I'll have in the talk today, because what I want to talk to you about is how computers are used to design new medicines. And as I say, we'll focus in on computer-aided drug design and molecular simulation. And we'll talk a little bit about artificial intelligence at the end. Okay. So I've said I'm a professor of pharmacy. I teach pharmacy here at the university. And just to sort of segue into the topic, pharmacists, one of the things that pharmacist students will study is drug discovery, where drugs come from. And some of that is to do with historically would have been just good luck. You probably all know the sort of story around the discovery of penicillin. But these days, it's often about drug design. We design drugs. And as I say, one of the things that I'm going to talk to you about is using small molecules to bind to bigger molecules to make a biological effect. And so some of those pictures on the side of the screen there are from my own research, and we'll talk a little bit about that as we go through the talk today. So a little bit of history. When I started doing computer-aided drug design, that was back in 1992. And again, just for just to get your head around what life was like then, the internet wasn't really a thing. Then it had only just started. If you can imagine the internet only just coming into existence. Back in 1992, there weren't websites to go and visit. I was doing my research in Oxford, and there's some pictures of what we call silicon graphics machines there. And in the lab I was in, literally sat there like that with a computer the size of a fridge sitting next to me. And that was the computing power that we had. Back then, we got a 1 terabyte disk from a very large research grant that 10 of us had to share, and it cost thousands of pounds. And now you can buy a 1 terabyte disk for £20. So things were a little bit different back then. I'm going to come back and forwards to think about kind of what the future might hold by looking back at the past a little bit. As I say, thinking about why we use computers and how we use computers for drug design. I mentioned, that luck, serendipity can often play a part. We've talked about penicillin, but also observations. So you might well know that aspirin came from the observation that if you chewed on the bark of the willow tree, then that acted as a painkiller. And it was only much later that we found out that, well, that's from salicylic acid that was present in willow bark, and then chemists made a variant of it, acetyl salicylic acid, and that is now what we take as aspirin. So serendipity, good luck, also making some observations. But what the invention of the computer did, it helps chemists not just to keep trying different things and see whether they work, but with computers we can predict what molecules might be good ones. So again, historically, the chemists would have had to make compounds and test them and hope that they got a good molecule. Whereas now with computers, we can do something called structure-based drug design. We can try and predict what molecules might be good, and then we can tell chemists of all the millions of compounds they could make, here's some which might be good, here's some which might not be so good. So how do we use computers to do this drug design work? Well, some of you might be familiar. I can't see my screen at the moment, so I'm hoping you can see me waving a molecule in front of me. If not, if it might be disappearing with my background. But some of you might be familiar with these kind of plastic-based models. And these plastic-based models help us to know where atoms are in relation to each other, and we can put bonds in. But we can only really do small molecules that way. You might have seen this picture here of the DNA double helix created by Crick and Watson. And again, that's pretty much about as big a molecule as we can get. And that's a hefty model to kind of be working with. But even then, that's only a few base pairs. It's not actually a very big model. So, but we can visualize models using plastic, using metal. But it's far easier now to visualize atoms in 3D using computers. So here's some screenshots of how we might kind of use computers to think about where atoms are. And these are all different ways of visualizing atoms and structures. So on that top picture, the sort of molecules all superimposed on the left-hand side, the kind of blues and greens and reds there. That's over here. Then these are kind of, each point there is an atom. We've taken away hydrogen atoms so they don't mess up the picture. But even then, that's still quite a lot of atoms. What we do in the middle picture is we show protein chains all linked together where the amino acids are all joined together. These are actually hundreds of amino acids. Each amino acid has 20, 30, 40 atoms in it. So thousands of atoms, but we've simplified the picture to try to work out our drug molecules stuck in the middle there. We can work out what sort of interactions it's making and see if we can make some better predictions. And again, on the right-hand side, we then kind of focus in on what bits of the molecule we're interested in. So even though we've got thousands of atoms and those pictures at the bottom, every dot on every line is an atom. So you're talking thousands and tens of thousands of atoms, but we try to break it down so we can visualize what these atoms are doing in 3D. And with computers being as good as they are now, we can perform fast calculations to try and predict where those atoms are in space. And then we can start asking questions. Well, what happens if I add an oxygen over here? What happens if I add a carbon over on the other side of the molecule? What happens if I change this bond? And we can begin to make better and better drug molecules. Now, the problem that we have is that knowing whether we're right or not, knowing where these structures come from. So using this idea of structure-based drug design, because we're designing our drugs based on a structure, that's what that title says, we need a structure to start off with. And again, if you're doing A-level chemistry at the moment or anything like that, you might have come across some of these topics as to how we know where the atoms are in space. We can do some experiments. We can do things like X-ray crystallography. We can do nuclear magnetic resonance spectroscopy. We can use, or we can, as they use the computers. But we tend to start off with a starting structure, maybe from X-ray, and we build on it. And we call this homology modeling. So once again, just looking at where we're at, this is a graph of the number of structures in a database called the Protein Data Bank, the PDB. So this is kind of known structures where someone's done an experiment and we know where the atoms are, and that's then deposited for researchers to be able to use. Now, again, down here, if you can see where my pointer is, let me just put a pointer on here just so you can see. So down here, this was about that time when I was doing my PhD, 1992, 93, 94, around this sort of time. Now, this is important because I'm going to show you a graph later on about where we're at with AI, but just look at the number of structures that were available when I started off on my research career, and look where we are now. They've gone up in this exponential fashion up to 2025, where we are now. So massive growth in the structures that we know about. Okay, so what about drug design? So when we're designing drugs, what we're trying to do, you might have heard of this idea of a lock and key concept. What we're trying to do is get our drug molecule to fit inside the protein. And normally it's either blocking the protein and stopping it from working, or it's coming in and doing whatever the natural ligand would do and doing it better. So we're either trying to kind of get the target to stop working or to work more efficiently. So a lot of drugs that we deal with, it's doing that stopping, it's that inhibiting the protein. So what we're looking for is this kind of lock and key, this idea that there's a drug that's the right shape that will fit neatly into that target. So you can see there's a nicely sort of fitting drug into a nicely fitting kind of target shape up here. And here is a space-filling picture of this. Green is Gleevec, a drug bound into its active site. You can see it fits really snugly into the active site there. If we've got gaps between the drug and the target, that means we've got weak interactions, and we could try and improve that if we look for those. Close fit is good. And if the drug is clashing with the protein, then that's a bad thing. That means we're going to get kind of repulsion, and that's not going to bind very well. So when we're doing structure-based drug design, we're trying to bring these ideas together. We're trying to make sure we've got a structure of our target that we're comfortable with, and then we design our small ligand to fit into it, the hole that it fits in. This is a piece of work that I did a few years ago. This was an anti-cancer drug, and the chemists knew this structure of EO9, but they didn't know how it was binding. So they were just going to try changing bits of the molecule. They were going to try changing these two ends. They were going to try changing these two oxygens in red over here. And they were going to try changing this kind of blue and green bit, this kind of triangle at the end here. And the chemists were going to start with the end bit, and they were going to try work this way. So we did some computer modeling and worked out how this was binding into the active site. And you can see this is like a cave. It's sort of sitting in this kind of cave-like hole in the protein, but it's gone in with this triangle bit first. And you've got to try and visualize that this TRP 106 is actually the back wall of the cave. So trying to change this sort of EO9 bit here, this triangle bit, would just push the molecule further out. And these two oxygens, these two red bits here, needed to be where they are so that they could do a reaction with this point FAD beneath it. So we looked at this and said, but if you look at what's kind of poking out the mouth of the cave, you've got this chain here that leads to B, and you've got this chain here that leads to A. And we could work out that there's an interaction here. So we said, well, that's already got a good interaction between A and this TYR 156. So we said, let's leave that. But this big long chain here, this is what we want to play with. So we saved the chemists a lot of time, because if they'd messed around with any of these other bits, it would have broken the molecule and it wouldn't have worked. Whereas this bit, we could make it even better. So that's what we did. So some of my other work has been a bit harder. We've kind of looked at structures which sit across membranes. If you've done any biology, you'll know about kind of cell membranes. And there's a class of proteins that sit across membranes, and those proteins called GPCRs, they do a lot of cell signaling. They do a lot of how the body responds to things which are working well, things which are not working well. And they're often a target for drug molecules. So they're much harder because there's far fewer structures available. So that graph of structures, in terms of membrane proteins, they're much harder to crystallize. They're much harder to get structures of. So what we wanted to do was try and use computers to design better drugs for this class of protein. So let me talk you through an example of one that we did. This particular class of proteins called the cannabinoid receptors. You've probably heard of cannabis. And cannabinoid receptors bind the natural compound in cannabis, and it has two effects. One is to relieve pain, the other is to give the illegal high. Now, what we want to be able to do was isolate that pain response, which comes from a particular protein called the cannabinoid receptor 2, CB2, whilst not giving the same sort of euphoric high effect that comes with binding to CB1. This enables us to then create a drug that can really target pain. There were no known structures of this receptor, so we went out and started building a molecular model. So we used the computer, we took the protein sequence of these two receptors, and we built computer models, which is shown over here on the right-hand side. We then found some known ligands. So we've got agonists and antagonists. These are things which bind to the protein. We know that. We've got some experimental data. And then we use them in our computer to say, well, do we get a similar sort of binding in our computer? And yes, we do. So now we've got a computer model that we're happy with, that we know can work. And then we start using our eyes to say, well, what's going on? We take some known compounds, we dock them into our model, and then we start doing that piece of work that I talked about, where we start saying, well, what if we change this? Where is there we can build on? What can we change that works? Which bits of the molecule do we know not to touch? And then we get the chemists to make us some new molecules. So that's what we did. We made some new molecules and we got some experimental data, and some of these are interesting and some are less interesting. But what it meant was we can use the computer to design drugs, so we know that we can get some interactions which are good, and we can improve on some of the drugs that are out there. So this was an example of where we'd taken a structure and we'd use the computer to be able to tell the chemists where to spend their time and effort and how to make better drugs. So what about the future? You'll have all heard about artificial intelligence. Some of you will have been using artificial intelligence models. Some of you might have been doing some homework with ChatGPT and other things like that. What about artificial intelligence in drug design? Surely this is something that we could do. And sure enough, there has been some activity in this area. So in the past decade, we've got a number of AI-discovered drugs that have come onto, well, one's come onto the market, but others are still in development stage. But what's important is the rate of advance. So here's a graph just showing back to 2023 and earlier. But what I want you to recognize here is not just some of the data in this graph, but do you see the shape of it? Do you see the shape of it going up in this exponential way? Do you remember when I showed you that first graph and said when I started my research career and the number of structures, it was very small numbers, but it was going up just like that. That's exactly where we are now with AI. It's very small numbers and it's going up, but this will grow and grow and grow. So what's this graph showing us? Well, we can use AI in the drug design process in a number of different ways. Firstly, we could use it to identify the target. So that's the kind of protein bit that we were talking about. Historically, that was done through scientists getting together and thinking, well, here's a target that could be interesting, and doing pharmacology on it and then saying, yes, it is, that is linked to this disease. So trying to find what the molecular target is linked to certain illnesses or disease states or conditions. We can now use AI to even pull out some of that information because AI is good at that. There are lots and lots of scientific papers with lots and lots of results. And it could be that at a human level, we just haven't made some connections to identify, oh, that disease state could be linked to this molecular target. AI would be very good at that and has been, and that's these red bars in this graph. So a lot more AI targets. What we tend to think about is, well, could they design small molecules in the same way that I was describing the way we've done it using computers? Could AI be doing that? Could AI be saying, well, let's try this with an OH on this part of the molecule. Let's try it with a methyl group on this side of the molecule. Let's try changing this carboxylic acid for an amine group and see what happens. So that number starts off very small. These are these blue bars, but as you see, is a growing component of where we're going. I think probably the vaccines and the kind of repurposing molecules, those will always be quite small numbers. Repurposing molecules is where a drug has been developed for a particular disease and someone is, you could then kind of use it in another disease state. I think those will still stay quite small, but identifying targets and identifying small molecules, you can see those bars increasing on size year on year. And in the scientific literature and in some kind of headlines, this is exactly what we see. So we're seeing an increase in the sort of pipeline of drugs, but we're also seeing drugs coming into clinical trials. So the problem with some clinical trials is historically we develop a molecule, it would get into clinical trials and then it would fail. The interesting thing about AI is it seems to be doing much better at picking winners than humans were. So it doesn't seem to be failing in those phase sort of 1, 2, and 3 trials as much as kind of human-derived molecules do. So that's interesting, and there's people doing work on that to try and work out why that is the case. But we tend to find that there's about 90% more success in phase 3 clinical trials than from molecules developed by humans. So AI does potentially have a use, and it might be that that's because AI is better at pulling out what makes a good drug molecule, what's more drug-like properties that we call it. So again, just to kind of give you a picture of where things are at in terms of some drug design, and so this is back to 2023. Say one molecule launched, a number in different stages of clinical trials, but that pipeline, again, just look at that pipeline going up, and that exponential growth. And again, some of that arising from those small molecules derived from AI. But it's not without its problems. And again, if you've thought about artificial intelligence or you've read anything about artificial intelligence, you'll know that ethics is a big issue in artificial intelligence. One of my family members this morning produced a thing on our family WhatsApp chat to say, here's my latest album on Spotify. Well, it wasn't his album. He hasn't got a musical bone in his body, but he got artificial intelligence to create what was clearly good sounding music. Except artificial intelligence doesn't create new things. It's not creative. It uses existing data, existing information, existing patterns. So creating artificial intelligence music will inevitably be using existing other people's creations. And there's ethics around that. In terms of drug design, the ethical part stems from the masses of data that is used to train some of these AI models. Now, if a drug company has invested a lot of money and effort in getting data to develop new medicines, it could be that their target market for those medicines are for people who have the money and societies that have the money to spend on those medicines. Which could mean, for example, that their data is very much skewed to the northern or the western hemisphere. It could mean that data is very focused around people of a certain ethnicity. And that could exclude other people. So the problem here is that the data that AI is training itself on could mean that new medicines end up targeted towards certain folk in society, and other people are excluded. So there's a whole set of ethical issues around focusing on particular populations and kind of the computer going in a particular direction, and that could become problematic. So companies like GlaxoSmithKline are spending a lot of time and effort to make sure that their systems and their AI processes are not biased because of the data that their AI is trained upon. Okay, so hopefully in the last half an hour or so, what I've done is kind of give you a little bit of a glimpse as to how computers can be used in drug discovery, about how I've used them in my own research for that. Computers certainly have been and continue to be used in this kind of rationalizing the drug design process. They can be used simply to visualize many thousands of atoms in proteins, but more importantly, we can use them to then design small molecules, to work out how those small molecules are interacting with the protein, and to then hopefully design better drug molecules. And we've talked a little bit then about how artificial intelligence is having a bigger impact on drug discovery. And again, hopefully, if you get into drug discovery and drug design, either through scientific research, you might be interested in pharmacy or medicines degrees, which we run here at Bangor. if you begin to kind of look at these things, then I'm sure that as you develop and you look back on your career in 30, 40 years' time, you'll see kind of again that similar growth of how artificial intelligence has impacted drug discovery. So that's a whistle-stop tour. I'm happy to take questions or queries. If you want to get in touch with us, do so, but hopefully you can use the QA chat function. I'm going to stop sharing my screen now.
Thank you, Stephen. So at the moment, there aren't any questions in the chat. So if anybody has any questions, feel free to put them in the chat. Or if you have any questions about anything, anything to do with our courses here at Bangor, or as well as to do with Stephen's talk as well.
Great. I can see a question there from Tasnim, is it? Yeah. Yeah.
Oh,
Fantastic.
sorry, I couldn't see that one.
Yeah. It's just come in. Brilliant. So the question is, do you think that the progress that AI is making will plateau at some point? Well, it's an interesting one. If you look at that data growth, and I was kind of trying to say, look, I think you're going to see something similar. That really is that exponential growth. I think we're going to see that happen. I think we're going to continue to see more molecules coming through in that kind of rate. What's the more interesting question is, are the tools going to get, what's that sort of rate of acceleration? You can have sort of exponential growth that goes at kind of a slow, or it can go fast. I think what's interesting is the risk of AI creating new molecules and new data, but if you haven't got that human creativity piece coming in, which we've had for decades and centuries, then you're just making new things based on old information. So that's the risk with any AI, even with the large language models and ChatGPT. It's based on scraping the internet at a point in time. If you then fill it with things which aren't working, then you're moving forward and creating more rubbish, and you're drowning out the creativity piece. So will it plateau? I'm not sure it'll plateau. Will it lose its effectiveness? Maybe. Unless something changes, unless there's some form of quality control put on where the data comes from.
Okay, so there's another question from Natalie. What would the university pathway for getting into drug design look like? Is there more than one route?
Yeah, so I'll answer the second part of that. Yes, there is more than one route. And again, it probably just depends on what where your interests lie. And sometimes, you can vary once you go down certain pathways. So my background is I started off life as a chemist. So I did a chemistry degree because chemistry is what I liked. I liked the small molecule side of the question. Soon I realized, having looked at chemistry, that actually what I was really interested in was this idea of drug discovery and drug design. And then I had to kind of realize that the small molecule fits into a big molecule. So I've now got to understand some biology, and I've got to understand proteins. And I kind of got into kind of drug discovery from the chemistry angle, then knowing a bit more about the biology. And now I teach pharmacy, which I'm now thinking, well, that drug is involved with the patient. So now I'm also thinking about the patient. Other colleagues of mine started off life being a pharmacist and thinking about the patient, and then being interested in the biological molecule and then learning the chemistry. Here at Bangor, we run a pharmacology degree. So again, you could, if biology is more your thing and you're not interested in the patient, but you want to do drug design, but you want to come at it from a biology perspective, then probably pharmacology is your starting point, which is more of the biology end of this kind of activity. So lots of different pathways into drug discovery, drug design. Our pharmacy degree and our medicine degree, I guess, focus significantly more on the patients. So we'd expect you to kind of go into those wanting to help patients. So be with a patient focus for medicine or pharmacy. But in either of those degrees, you could then later on go, well, actually, the science bit, the drug discovery bit, the kind of is what I want, is where I want to go. And people do that. They change careers, go into more the science side than the patient side. But that would be a bit of a strange route. You'd have to want to start by having an interest in patients. If you know it's the science, then have a think about chemistry, have a think about pharmacology, as good routes into drug discovery, drug design.
Okay, thank you. So are there any other questions before we wrap up this session? Plenty of time to put them in the chat quickly. Okay, so that was a fantastic talk. Thank you, Stephen, and thank you for all your questions. So we hope you enjoyed today's session, and if you're interested in pharmacy or any of the other North Wales Medical School courses, please take a look at our web pages or join us at one of our open days this summer.